So, welcome to Biomindscape. Today, we are going to know a new topic, that is transplantation. It is a process of taking a tissue or an organ, that is called a graft, and we have to place that from one individual to another, and this individual will provide the graft, that is called the donor. So, an individual who receives the graft, it can be either recipient or the host. So, blood transfusion is also a form of transplantation, in which the whole blood or a blood cell from one or more individual are transferred into the circulations of the another individual. So, immune system have evolved as a way of discriminating between the self and the non-self. So, it has a power of selecting a undesirable or a desirable character in a particular tissue and organ to accept it or to reject it. So the tissue and organ it is transplanted from one individual to another for therapeutical purposes. So transplantation of a tissue or an organ from one individual to a genetically non-identical individual leads to rejection, that is called the graft rejection of the transplant due to an adaptive immune response. So some transplanted tissue does not stimulate an immune response. For example, a transplanted cornea is rarely rejected because of the lymphocytes, and it do not circulate into the anterior chamber of the eye. So this site is considered as immunologically privileged site. So another example of privileged tissue is heart valves. So let's know about different types of transplants. So autogenic, syngeneic, allogenic and xenogenic. So what is an autogenic grafting? A graft or a transplant from one body part to another on the same individual, that is autogenic. Then syngenic is a graft transplanted between two genetically identical individuals, that is a syngenic. In human, an syn graft can be performed between the monozygotic twins. Then allogenic grafting, it is a graft transplant between two genetically different individuals of a same species. And xenogenic grafting is a graft transplanted between individuals of different species. So autogenic and syngenic are usually accepted owing to the genetically identical between donor and recipient. So because of an allograft is genetically dissimilar to the host and therefore express unique antigen, so it is often recognized as an foreign by the immune system, so it and is therefore rejected. So xenogenic graft exhibit the greatest genetic and antigenic variation, causing a vigorous grafting rejection responses. So today we are going to know about the xenogenic transplantation. It refers to the transplantation of living cell, tissue and to another, and most commonly they are found in animals and humans. So this xenogenic transplantation is a process of grafting, of transplanting organ, tissue or a cell from a donor of one species into a recipient of another. So xeno means a foreign or different. This transplantation is interspecies, that is like pig to human. That means organ is transplanting from an animal to a human being. So this type of xenogenic transplantation, it can be cellular, it can be tissue type or organ. So the cellular xenogenic transplantation, in this transplantation of an individual animal cell like pig pancreas, isolate cells, these are transferred into human with diabetes. Like tissue transplantation, in this transplantation of a tissue like pig heart valve, they are transplanted into the human heart. Then organ xenogenic transplantation is like the whole organ like pig heart or kidney into a human patient. So it is an experimental trait. So why it is used in animal like pig? Similar organ size and physiology, because it has a similar organ size. It is easy to breed and genetically engineered. Short reproductive cycles. They are already used in food and biomedical industry like insulin, heart valves, etc. So, what are the challenges in xenotransplantation? Immune rejection. The human immune system attack the foreign organ like hyperacute rejection or cellular rejection. Zoonotic disease transmission, in that the risk of animal virus infection infecting the human. Example, the PERV, that is the porcine endogenous retrovirus. Then it have some ethical concern like animal rights objections and concern over human animal genetic mixing. Then physiological incompatibility like difference in the blood pressure, enzyme function, etc. Then advancement to overcome these challenges, they have immunosuppressive drug to reduce the organ rejection, screening animal for pathogen, use of humanized pig like genetically modified to express the human protein, or using a CRISPR case to knock out the antigenic gene, or genetic engineering of a donor animal. So these are the some of the advancement to overcome the challenges. And the current use is that experimental the pig heart and kidney transplant in critical human case, example in experimental surgeries in the US, and pig islet cells for diabetes, and it is used for Parkinson diseases. So the regulatory bodies worldwide are like World Health Organization, FDA in US, and ICMR in India, and guidelines are strict due to safety and ethical issues. Thanks for watching. Do like, share and subscribe.